It was at that, I hosted for the biology department at the open house last night, and it was nothing but Zoom problems. Very exciting. Zoom issues. 28, vote, vote, vote. 7 more. Come on, pup. Five more. Cast your votes.
No, I can't see the poll. I didn't even know you sent it out. That's good.
Three more people. Vote, vote, vote. Two more. Okay. All right. Last call. Going once. Going twice. All right. So we talked about organic molecules last class, right? We started going through the 5 organic molecules have a carbon backbone. Remember, organic means you have carbon in the body of the molecule. So we went through hydrocarbons, carbohydrates, lipids, proteins, nucleic. Oh, well, we're going to go through proteins and nucleic acids today. And we talked about how for each category of molecule, you need to know the properties of the molecule, the component monomer, its biological significance, and be familiar with the examples we talked about, right? Oh, did we not talk about, we didn't talk about proteins yet? No. All right. So good job for those of you guys that are ahead of the game. Monomer for a protein is amino acid. I think I mentioned it a couple times in last lecture, but we'll talk about it a little bit more. Again, monosaccharide was the monomer for which category of organic molecule? Anybody remember? What's monosaccharide the monomer for?
Carbohydrates.
Carbohydrates, yeah. Fatty acids is not a monomer. So we left off last class talking about lipids, and remember that for lipids, we said that's the oddball category, right? Where it doesn't have a component monomer that builds it. Some of them will have fatty acids, but not all of them, right? So it's not a actual monomer. Amino acids, we're going to talk about today, how they work and how we build proteins. And then we'll wrap up today with nucleotides. So for nucleic acids and nucleotides, I'm just going to give you a little bit of information and introduction. I'm going to talk about that category of molecule in greater depth. Did that show? Did the results show for you guys? I'm getting weird multiple boxes popping up. Okay. So nucleic acids, we're going to spend a lot of time talking about in Unit 2. So I'm going to give you just the basics for nucleic acids, and then we'll come back to them next unit, starting next week. Ooh. All righty. So reminder, hydrocarbons, right? The least complex of our organic molecules made up of only carbon and hydrogen. We said they're very important for energy sources, for communication, and as the building blocks for building some of our other organic molecules. So we talked about how you can swap off a hydrogen and add a functional group in order to change the category and the property of the molecules of hydrocarbons, right? So functional groups we're going to see in all of our types of molecules. They are clusters of atoms that are reactive and influence the property of the molecule. So we'll talk a tiny bit about functional groups some more today. Our second category of molecule, carbohydrates, right? So we've got to learn about monosaccharides and how there's a whole variety of monosaccharides, the monomer for carbohydrates. We talked about how you can put monomers together to build polymers, right? So we talked about disaccharides, monosaccharides, and disaccharides. Dietarily, we usually refer to as simple sugars that are quick, easy access to sugar molecules for energy purposes, versus complex carbohydrates, which are larger chains of monosaccharides. So we talked about how our polysaccharides are really important storage molecules or structural molecules. If you're talking about cellulose, plants use it to gain rigidity to their tissues. And we'll talk about that a little bit more as we start learning about cells, hopefully at least a little today. Okay, we left off learning about lipids. Lipids, we said, are categorized not by a typical monomer, but by their property of being, they're all going to be nonpolar. In other words, they're all going to be hydrophobic. They're not going to interact well with water. All right, so we were on our way through learning about the diversity of lipids. We talked about how they can be used to insulate bodies. We talked about how they can be used as energy sources. So we learned a little bit about dietary fats and talked about the difference between fats and oils. We talked a little tiny bit about the health implications. And this is where I left off, right? We're going to learn about waxes and a couple other types of lipids before we move on to learn about proteins. So waxes are going to be really important for physical protection, so they can help protect tissues against abrasion. They can also be used for protection against evaporation, right? So remember that our bodies are largely made up of water, and water can be a limiting resource. So you don't want to be evaporating it off the surface of your body all the time, right? You want to waterproof the surface of your body so that you're not losing all the water and having to constantly be replacing it, right? Organisms are going to be producing waxes and oils to help waterproof, right? So plant leaves will do this, right? So plants will oftentimes have a waxy coating. Their fruits will have naturally waxy coatings, not as thick as what you find at the grocery store, but they will have wax, a thin layer of wax. Animals will use waxes. So some birds, for example, have waxy secretions that they're going to put on their feathers to waterproof. Waterproofing is super important in mammals as well. So that sea otter needs to constantly be either putting a waxy substance or oily substances on their fur. That's going to help to waterproof it so that the cold water doesn't make contact with their skin. It provides a little insulatory layer of air in there. So really important for waterproofing, yeah, and protecting against abrasion. Do we produce wax? Yeah. Where in your body do you produce wax?
Our ears.
Our ears.
Mm-hmm.
Yeah.
And you know what that's for? Not to gross you out. It's for waterproofing your ear canal, right? It inhibits water, repels water from collecting in your ear canal and causing problems, right? Creating a moist little habitat for bacteria or fungi. Right? So wax, super important. Okay, so phospholipids. Phospholipids are going to be really important category of lipids. They're going to be the building block for cell membranes, right? The edges, boundaries of our cell. I'm going to talk about phospholipids a lot more later on, but I'm going to give you an intro today. So phospholipids are these crazy molecules that are sometimes illustrated like this ball with two little legs, or what's called a tail. It kind of reminds me of like a little hairpin, right? The way that they're illustrated, but keep in mind that they are a complex molecule, right? And the molecule has this really interesting amphipathic property, right? And what that means is that it's dual natured, right? One side of the molecule, what we call the head, right, is polar. Right? And the other side of the molecule that has these two fatty acid chains is nonpolar. So this molecule is partially polar and partially nonpolar. Right? It's partially hydrophilic and partially hydrophobic. Right? And the reason why this is important is because half of this molecule is going to interact well with water and half of it is not, right? And that's going to drive the behavior of this molecule. When it finds itself in water with other phospholipids, it's naturally going to align itself. They will naturally align themselves in this 2-layer of phospholipid structure, right? It's called a bilayer. The phospholipid bilayer, if you take a look at how it forms, right? Notice how all the hydrophilic heads, the polar regions, are going to face in one direction, and then down here, all facing in the same direction. And they're going to point their tails towards each other to make this hydrophobic zone. So anytime they find themselves in water, right, or in a watery solution, they're going to align themselves into this bilayer, right? And what that does is it has all the hydrophilic heads facing the water here on this side and on this side down in here, right? And the tails are protected. So by doing this, just because they're interacting with, the water around them, they make a boundary. And so phospholipids on early Earth, right, started to aggregate and make these little bubbles, created the precursors to cells. And they will align themselves like this on their own. You don't have to spend energy to put them in this structure. So phospholipids are going to be the basis of our phospholipid membrane, the membrane of the cell. We'll come back and talk about them a little bit more. Your cell membrane is more complex than just phospholipids. It's going to have a whole bunch of other molecules embedded within it, but the phospholipid is the main meat of your cell membrane. Make sense, phospholipids? Any questions? Really cool molecules. All right. Next, I'm just going to mention steroid hormones briefly today. I'm going to spend more time talking about hormones and how they work when we cover reproductive systems. So I'm going to kind of reproduction and sneak in some endocrine system into that lecture. But steroid hormones are lipid-based, right? They're based on a cholesterol molecule. So if we take a look at, check out this cholesterol molecule illustration at the top here, right? And when you look at testosterone and estrogen, can you pick out what's different, right, between cholesterol and these two hormones? So notice at the top, right, this functional group, and then half the time this functional group is different. Ooh, so I can turn a cholesterol molecule into a steroid hormone just by clipping off and swapping out one functional group. Functional groups really important, right, for changing the properties of a molecule. It's going to change its shape. It's going to change potentially the reactivity of the molecule. So if we take a look, we can clip off this big functional group up here and then use that cholesterol molecule as the basis to make your sex hormones, right? And then within the two types of steroid hormones, notice that the difference there is the location of this double bond, right, is in separate corners. And that's the only difference between testosterone and estrogen, right? They're very similar otherwise. And indeed, in the male body, he's going to use excess testosterone and convert it into estrogen as the source of estrogen. Right? So those two hormones are incredibly important for building and regulating bodies, right? They have a whole variety of different jobs that I'll talk about when we get to reproduction system. Make sense? Ooh, just a little bit of difference in shape, right, can have a big impact on the body, right? So testosterone and estrogen, again, very, very similar shape. Just one location of a functional group is what's different between them, right? And it can have a very different impact on the body. Shape, so important in molecules. Cool? Okay. So proteins. Proteins are a really important category of molecule. We said our bodies are about 65 to 70% water. The reason why you're not sloshing around is because you have a bunch of protein emulsified in there, right? Proteins are going to be incredibly important in all the different jobs that they perform in a body. So what we're going to do is talk a little bit about the diversity of proteins jobs. And then I'm going to talk about how we build proteins, right? And proteins, again, we're going to come back and talk about when we learn about gene expression, we're going to talk even more about how we build and code for proteins, right? It's a really important job and one that we happen to understand fairly well in terms of DNA expression. All right, so proteins we said are going to be using that monomer amino acids, right, to build these large, complex, three-dimensional molecules. And again, shape is critical, right? Shape is going to determine function. And the diversity of shapes that you can build with proteins is going to allow all the diversity of functions. So in a little bit, I'll talk a little bit about how we put amino acids together, right, in a long chain, and then we fold that protein, right? It's going to spiral and have some of that pleated sheet that carbon naturally is going to form, and then we're going to see complex folding patterns in that protein. So in my crazy mind's eye, proteins are kind of like making an origami, right? They're folded in just a specific way. Now, if the protein doesn't fold properly, it can't function because it will have a different shape, right? It's like if you made a mistake folding your origami swan, right? You would end up with a swan with no head, maybe, right? And it wouldn't be able to work, or a swan with no wing. So proteins, let's first talk about the diversity. Proteins can be used structurally for building things. So a couple of the really important proteins that we're usually familiar with in our bodies are keratin and collagen. Keratin protein is protein that's added into our cells. Keratin protein is it's kind of hard. It adds a hardness to your tissue that's protective against abrasion. And it's also a waterproofing protein. It's hydrophobic, right? So if we think about it, we have keratin in our skin cells, right? That's what makes our skin a little bit hard, right, and dry. The same type of tissue that makes your skin is the same type of tissue inside of your cheek, right? So when I say it's hard and drier, right, your skin has a different consistency than the inside of your cheek. The difference, the skin cells have keratin added to them. The inside of your cheek, no keratin in those cells, right? So you can see the keratin hardens it a little bit and makes it waterproof, right? So you can reduce how much water you're evaporating off of the surface of your body. Make sense? Right? The more keratin you put into your cells, the harder it gets, right? So if you put even more keratin in the same type of cells, you get hair. Oh, harder than your skin cells. Right? If you put even more keratin in, you can get a harder structure like a fingernail, same type of tissue. Right? All stratified squamous epithelium, right? But how much keratin you put in there is going to change the property, right? So a lot of keratin can make a hard structure. And your fingernails are there to help you, you know, to pick little ectoparasites off your mates, and also helps to protect the tip of your finger. Right? If I added even more keratin, I could build a horn or a shell, like on the outer part of a shell of a tortoise. Make sense? Collagen, on the other hand, the most abundant protein in your body, right? It's found everywhere, in between your cells, as well as building tissues like tendons and ligaments have a ton of collagen in it. Collagen, really important for attaching structures together. They're really, really strong. But anyways, I'm geeking out on proteins. You get the idea? Structurally, they can be important, right? So, for example, tendon is going to very strongly attach a muscle to a bone. All right. Protein can be used for storage, right? So you can store amino acids in a legume, right? Protein also has energy in the bonds that are between the amino acids and within the amino acid. So when we're eating things like legumes, right, we're eating the protein that's in there. And the protein is, you know, in part part of the little embryo and the resources for the embryo once it starts to germinate. So storage, protein can be a storage molecule. Protein's incredibly important for motion, right? Motion at all scales, for those of you that have watched the Pattern of Science video already. So if we take a look, maybe for Microscope Lab, you got to see some microscopic images. If you take a look, here we're looking inside a plant tissue, right? And these are all plant cells. And inside, you can see all the little organelles moving around, right? And that's because inside of your cell, you're actually circulating the solution that's in there around and around and around to distribute oxygen and nutrients all throughout the cell. That motion is due to proteins that are in there creating that current. Within your cell, the way that you move structures like organelles around, if you're dividing or transporting something, is using these little crazy proteins called kinesins, right, that walk along the cytoskeleton, also made out of protein. So any type of motion that you're producing is due to proteins, right? Inside your muscles, it's proteins sliding against each other that are causing you to be able to contract a muscle and move your body, right? Every scale of motion, thank you, proteins, right? So creating motion, but also transporting things. We're going to talk about membrane proteins quite a bit next week. So membrane proteins are protein doorways that are embedded in your phospholipid bilayer of every single one of your cells. And it's in a large part, one of the ways that you move things in and out of your cells is through these transport proteins that are embedded in your cell membrane. So you can move things in or move things out. In your bloodstream, on your red blood cells, your red blood cells are coated, right, in hemoglobin, which is a protein. It's there to help bind to gases so that you can transport gases to and from your lungs and your tissues between those areas. Every single one of your cell membranes has labels, protein labels, right? They're signaling to indicate the difference between a heart cell and a liver cell and an intestine cell. Every category of cell has a different protein label that helps your body to know, right, what's a liver, what's a heart, right? So if you're transporting something to your heart tissue, that little molecule is looking for that heart protein marker that tells it this is the cell that I'm supposed to go into, right? So every type of tissue has a different marker. The other thing that's really cool is that we also have markers that are unique to us, right? So I always joke, every single one of my cells in my body has a Letitia label, right? It's a specific protein that's only marker that's only found in my cells, right? No one else has a Letitia label on their cells. You each have your own version of labels for your cells. And it's a really important way that your immune system works, right? So we have what's called an acquired immune system. It's acquired as you are exposed to the world. So you're born without it and it develops in your first few years. Part of what's happening as your immune system is building, your acquired immune system, is that your immune cells are going around and introducing or learning to recognize your markers of self. So when my acquired immune system was forming, my immune cells went around and I call it introduced themselves to all my cells. Hello, Letitia heart cells. Nice to meet you. See you around the neighborhood. Hello, Letitia liver cells. Hello, Letitia kidney cells, right? And you learn what cells are yours. And that's really important part of your immune system, right? Also made out of proteins, because those immune cells, as they're patrolling around your body, they're like, okay, that's Letitia heart cell, that's Letitia liver cell, those cells belong here. When your immune cells come across a cell that doesn't have a marker of yourself, right? So in my body, if my immune cells came across a cell that didn't have a Letitia label, the way your immune system works is that it goes, oh, you're not self. I didn't meet you in the neighborhood. I'm going to attack you and kill you because you don't belong here, right? So if you get a pathogen, right, like a bacterial infection, those bacteria cells aren't going to have Letitia labels. So my immune system's going to attack it. Does this make sense? Kind of cool. So what happens when you have an organ transplant? And now all of a sudden I have a hunk of Bob liver in my body with Bob labels. What do? You have to take immune suppressors. When you've had an organ transplant, you have to forever take immune suppressors to prevent your immune system from attacking those cells that don't have your label. And sometimes it doesn't work. And this is what we call organ rejection. When your body's like, no, absolutely not. That's a Bob cell. It does not belong in this body. Right? And out you go. Right? Make sense? Proteins. Ooh, so important. So what happens if your immune cells develop amnesia? Stop recognizing your own cell markers.
It might attack your own cells.
You will attack your own cells. This is what's called an autoimmune disease. Right? When your immune cells lose the ability, for whatever reason, to recognize your own cells, and you start attacking and killing your own cells because you've forgotten that they're part of you. So proteins, I can't emphasize how important proteins are. Right? The last function I'm going to talk about for proteins is the function of being, some proteins are going to be enzymes. Right? Some proteins are going to be acting as enzymes. And enzymes are fundamental to life. Right? You would not be able to afford your chemical reactions without the presence of enzymes. Right? Some of your chemical reactions would never occur without an enzyme for it. So, excuse me. Let me tell you a little bit about how enzymes work, right? So enzymes are referred to as molecular catalysts. Remember that when you have a molecule in your body, it has bound, so all the elements gain stability by sharing or transferring electrons. They're stable. They don't want to break apart. It's going to take energy to break that molecule apart. Right? Energetic energy, or cellular energy, pardon me, ATP, usually. Right? And that can be really expensive, right? To be running all the chemical reactions that keep you alive and having to pay for it. Right? So we use molecular catalysts because what they do is they make those reactions cheaper. Right? So if we take a look, so remember that enzymes are proteins, so they're complex macromolecules made up of atoms. When we teach bio students about enzymes, we usually use really simplified, or molecules, I should say, simplified diagrams, right? So these nuggets represent this complex protein. Right? And when we take a look at a protein or an enzyme, in this case, this enzyme right in here, notice that it has this little divot here. It's called the active site. And remember what we said about how proteins or any molecule in your body functions is based on shape. Right? So the way an enzyme knows the molecule it's supposed to break down is based on shape. Each enzyme is specific to the molecule that it's going to break down. And the molecule that an enzyme breaks is called its substrate. So the substrate is the molecule that's going to get broken apart. Right? You want to break it down to its component monomers. You want to undergo hydrolysis, right? And so what happens when you make an enzyme is it's moving around in your cell and it's bumping into molecules, right? And it will only bind to the molecule that fits in its active site. It's recognizing it by shape. And once it does, right, we'll watch this little animation, one more loop. Notice how when it fits its substrate, see how it kind of squeezes, right? It bonds to it. And it kind of, I imagine it gives it a little enzyme hug, right? And that enzyme hug oftentimes will put strain, like by bending the substrate. And it puts stress on that chemical bond, so much stress that now it's only going to take a little bit of ATP energy to break them apart. Yeah. So it lowers the activation energy, the energy that it takes to break that bond, oftentimes by putting physical strain on it. Make sense? right? So let's take a look at the cycle of an enzyme, right? So here's an enzyme, and notice its substrate here, sucrose, right? Again, they're specific to each other, right? There's an enzyme for every single molecule that you want to break down, pretty much, right? And the enzyme is going to be named after its substrate, right? So if this purple enzyme here is, its job is to break down sucrose, we're going to call that enzyme sucrase, right? So anytime you see that ASE ending, that's a suffix that means enzyme. And it's telling you it's an enzyme, and the first part of the term, right, is the name of the substrate. So sucrase is going to have an active site that's specific to the substrate sucrose. What do you think we call the enzyme that breaks down lactose?
Lactase.
Lactase. The enzyme that breaks down maltose is going to be called maltase. ASE ending tells you you're looking at an enzyme. All right. So when they randomly bump into each other, right, and bind, the sucrase is going to put strain on that bond so that you only have to invest a little bit of ATP energy, right, to break it apart. And once it breaks the substrate, it releases the parts into the environment. And now you can use that glucose to cycle more ATP. Right? And notice what happens to the enzyme. It's free to go turn around and bind with another substrate. So the enzyme isn't used up or damaged in the process of breaking a molecule, a substrate. It's going to be used again and again and again and again. And as long as its active site is free, right, it will continue to break down its substrate. Part of how we can regulate the activities of an enzyme is by blocking the active site. And you can do this in your cells naturally. And this is also how many medications can work, right? Is by being a similar shaped molecule that'll fit, right? And block the active site. And if it blocks the active site, the enzyme can't work. It's been turned off. So we can regulate the activity of our enzymes. Make sense? So they make your reactions cheaper and therefore also make them faster. Shape, 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 shape. All right. Okay. Lactose intolerance, I'm going to skip. I'm going to talk about that. I've got a homework assignment for you guys for lactose intolerance that I'll send your way later. Hey, get out of there. Get. He tricks me every morning. You think I would learn. He's like sleeping so calmly, and I don't want to wake him up and put him under the table. And as soon as I get rolling in lecture, he gets naughty. Hey. Oops. Here. This bark box came yesterday. Keep him busy. All right. Are you ready for amino acids? Woohoo! Structure of amino acids. Okay. So the amino acid is the monomer, right? And if we take a look at the amino acid, and you don't need to learn the chemical formula, but notice how it has an amine group, it's got a carboxyl group, and it's got that carbon backbone. Right? This green portion of the amino acid is the same in every single amino acid. Exactly the same. They all have that same basic structure. What's going to be different is this side group, or it's sometimes called an R group. There's one functional group, the side group or the R group, that is different amongst your amino acids. And there's 20 different side groups that you can use, right? So there's 20 different types of amino acids, right? The R group, the functional group, can have different properties depending on which one you're looking at. Sometimes they're polar, sometimes they're non-polar. Sometimes they're going to form ionic bonds, sometimes they're going to form covalent bonds. Right? The R group is the functional group. Right? And when we attach proteins together, right, we're going to be putting neighboring functional groups next to each other. And you don't need to memorize this chart, but it's a chart of our 20 amino acids. And what you'll notice is the amine group, the carboxyl group, the carbon backbone is the same in all of them. And what's circled in green, right? Oh, those, right, are the R groups or the functional groups. And notice how some of them, this one's carbon and hydrogen, right, only, non-polar. Whereas this one over here has an oxygen, so that tells me it's polar, right? They can have different properties. They're all different, the functional groups, right? And their properties are going to drive how they react. We'll come back to that in a second. When you build a protein, right, you use dehydration synthesize, and you get what's sometimes called a polypeptide, the long chain of amino acids. And the bonds between amino acids are sometimes called peptide bonds. You don't need to worry about that term for our purposes, but in case you're going on, you want to know what a polypeptide is. All right, so here we can see, right, different amino acids, right, coming together by dehydration synthesis to make a long chain. Right? And again, there you can see the OH, the dehydration process happening. Yeah, dehydration synthesis. Now, when we put that long chain of amino acids together, we call this the primary sequence or primary structure. So the primary structure is that process of putting together a long strand of amino acids. That's the first step in building a protein. Primary structure is the sequence of amino acids in a strand. It is the most important step. Again, it is the most important step because it's determining the sequence of amino acids in order is determining how the functional groups are going to interact with each other. In other words, how that protein is going to fold. Right? The primary structure is critical. The order of amino acids is going to determine how the protein folds. If you get the sequence wrong, your protein is not going to fold properly. And remember, if your protein can't fold properly, it cannot function. Big problems can happen. So primary structure is the order of the amino acids in a strand. It's the most important aspect of building a protein. If you get that wrong, you're in trouble. Yeah? A significant amount of the code in our DNA is dictating the sequence of amino acids. We'll come back and talk about that when we get to gene expression. So we will revisit protein structure. Right? For now, let me finish telling you about how a protein forms. Right? So once you put together your long chain of amino acids, your primary structure, then it's going to naturally start to get three-dimensional shape, right? The pleated sheet, if you guys remember this, remember when you attach one carbon to another, to another, to another, they naturally stagger. So it automatically gives you three-dimensional shape to your primary structure as those amino acids kind of stagger a little bit, creates that pleated sheet. And then you're going to have hydrogen bonds that are going to start to form on their own, right? That's going to cause parts of the protein strand to spiral, right? Like a curled ribbon. This is called secondary structure. You don't have to do anything. It's going to naturally happen once the primary structure forms. Cool? All right, so the part that is sometimes hard to imagine is the tertiary structure. So remember we said those functional groups, the R groups, are going to interact with each other. Right? A polar and a polar are going to be attracted to each other, and they're going to cause that protein to fold towards, to pull those two side groups together. If you have a polar next to a non-polar, they might be repelled and fold in the opposite direction. So which side groups interact with each other and how they bond with each other is going to cause the protein to fold. Right? So for example, maybe I have a functional group here and here, right, that are going to form a covalent bond with each other. And over here, I I've got an ionic bond maybe forming that's holding this loop together. And between these guys, there's bonds that are forming that are holding that branch in place. All the folding is held together by chemical bonds that are forming between the side groups. Yeah? So let me show you a picture. There it goes. Okay, so remember, here's your primary sequence. You've got, in this case, 3 amino acids next to each other. Notice how their R groups or functional groups are facing off to one side, right? As you build your nice long chain of amino acids, then you're going to end up with, look at that picture move. It's so bizarre. I call it the PowerPoint gremlins. I literally used this PowerPoint yesterday and those pictures were not overlapping. So bizarre. Okay. So as you build your nice long chain of amino acids that you can see here, right? Secondary structure is going to be the pleated sheet, the spirals that are going to naturally form. Tertiary structure is when the functional groups start to interact with each other, right? And maybe they're going to form ionic bonds, or maybe they're going to form covalent bonds, right? And those attachments between them are what are going to hold the three-dimensional shape to the protein. It's like the folds in your origami. Yeah? Mind-blowing, right? Okay. Once in a while, we're going to have quaternary structure that you see down in here. Some proteins, not all. And what quaternary protein is, protein structure is, if you notice, see the 2 shades of gray in here? Quaternary structure is sometimes in proteins, you have subunits that fold individually and then come together to work as a functional group. Right? That's quaternary structure. When you have 2 proteins that form by primary, secondary, tertiary structure, and then their last step is to come together and bond to each other. Right? Quaternary structure. We have a really important example of quaternary structure, hemoglobin. Right? The hemoglobin that's on your red blood cells to help transport gases is a great example of quaternary protein structure. It's 4 little subunits that come together. 1, 2, 3, 4 to work. Now, here's the interesting thing, right? Again, your primary sequence is the most important part. It's the most important part. If we take a look here, here's the representation of the sequence of amino acids in the coding for the protein hemoglobin. Right? It's got 146 amino acids in a chain. And then it's going to undergo secondary structure, the spiraling, the pleated sheet, and then, tertiary structure, bonds forming between the functional groups to fold and make that hemoglobin that's then going to coat the surface of the membrane of your red blood cell. Notice, right, if you have a mutation, a change in amino acid sequence, and notice how down here you swapped out 1 amino acid has mistakenly been replaced by valine here. Just 1 amino acid difference. And now that hemoglobin protein cannot fold properly and the red blood cell cannot form properly as a consequence. Right? And you end up with sickle cell disease. 1 amino acid difference. That primary sequence is really, really important. Right? If you change it, right, then you might not be able to fold properly and it can disrupt your function. To the point where if you take a look at sickle cell, right, see how the cell is a sickle shape, it's kind of hooked. These cells start to clasp onto each other, hook into each other, and make blood clots. Oh, massive blood clotting. Good? Not good. Deadly, in fact. 1 amino acid difference. So the sequence of amino acids, the primary structure, super, super important. Yes? Did I drive that home? All right. So again, what's holding the protein together in its 3-dimensional shape are all the bonds between the functional groups, that tertiary structure. Anything that breaks those bonds is going to cause the protein to unravel. Right? Anything that breaks the bonds holding together the different functional groups is going to cause the protein to unravel. And disassembling a protein is called denaturation. Right? So denaturation can be caused by a variety of things. Right? So temperature, for example. Right? If you get too hot, remember what heat is. Your atoms are agitated. And as you get hot, and you start agitating, eventually you're going to be agitating enough to break the bonds. And your protein unravels. So when an animal overheats, proteins are denaturing. Wah wah, X's in your eyes. You can also do the same thing if you get too cold. Your proteins can start to denature. X's in your eyes. When we cook food, like protein, the reason why, like a chicken breast, right? The reason why humans started cooking food is because it kills off any bad pathogenic bacteria that may happen to be in the food, but it also partially denatures the protein. So it's already breaking apart. So when you get it into your GI tract, there's less digestion that needs to happen. Remember that digestion in your GI tract is breaking the chemical bonds in your macromolecules, chopping them back down to their component monomers. Now, if you cook your meat first, then it's pre-digested. Then you have to spend less energy to break those macromolecules down to their component monomers. Yeah? This is the irony of the, what's that diet called where they don't cook anything? The raw diet. You're spending way more energy to digest your food. Why that's thought to, better digest your food? I don't know. It's actually the opposite. All right, temperature makes sense? Humans are a little bit crazy. All right, pH. Remember what we learned about pH, right? If you're acidic, you have a lot of lonely H+ ions. If you're alkali or basic, you have a lot of lonely OH ions, and they start interacting with existing molecules, right? So if you have an acidic solution, all those H+ ions start to interact and disrupt the bonds between functional groups and can denature that protein, right? So when you consume a protein in your diet, remember the pH in your stomach, 2, right? All those H ions start to attack and denature the protein that you just consumed. And indeed, protein digestion begins happening in your stomach, right? The acidic pH, but also some enzymes that you introduce there. Yeah? So my favorite example is if you've ever made ceviche, right? It's raw fish. Well, it's equally as denatured as if you had cooked it on the stovetop. The difference is you take that fish muscle protein and you squirt and soak it in lemon. And when you soak it in lemon overnight, all the H ions, the H+ ions, attack the protein in that fish and denature it. It also creates an inhospitable environment for pathogenic bacteria. It kills them off. Does the same thing as cooking it on heat would. Does this make sense? Look at how things are already starting to come together. Application of pH there. All right, and then we also talked about how denaturation can happen if you have a mutation, which changes the sequence of amino acids. Cool? All right, you guys familiar with cooking lobster? At least maybe seen it on TV, right? So here's what lobsters look like in nature, right? In their carapaces, their shells, they have a protein that gives them this bluish green color, right? And when you heat them, dump them in boiling water, they turn red. If you notice, the red's already in there in the carapace, right? It's just masked by the protein that covers it with blue and greenish colors. So when you put the lobster in boiling water, that heat denatures all that blue-green protein and exposes the red that was underneath. Yeah. Denaturation. All right. So last organic molecule. So far we've talked about hydrocarbons, carbohydrates, lipids, proteins, and our last macromolecule is the nucleic acid. So nucleic acids, there's a whole bunch of them, types that we're not going to talk about. I'm just going to talk about deoxyribonucleic acid and ribonucleic acid. I'm just going to introduce two of the nucleic acids. These two nucleic acids, their primary job is going to be to store and implement the information, right? So they're kind of like considered to be information storage molecules. They're going to have instructions for, your DNA is going to have instructions for building proteins, amino acid sequence. It's going to have instructions for how to regulate the activities of your cells. It's got all kinds of instructions built into it that help to keep you alive, right? Sometimes called, the blueprint for life. Nucleic acids are found in all living organisms, right? In eukaryotes, organisms that have a nucleus, the nucleic acid is going to be found inside the nucleus where it's nice and protected. Prokaryotes, like bacteria, are going to have nucleic acids, right? It's just not in their nucleus, right? So bacteria have DNA, right? So there's two forms of nucleic acids that we're going to be talking about: deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. They're abbreviations. Right? Both of these are going to be polymers of the monomer nucleotide. Right? So you're going to have nucleotides. A whole chain of nucleotides is going to help us to build RNA or DNA if you have two strands of nucleotides. For the nucleotide, the monomer, right, let's take a look at its structure a little bit. Right? You've got a carbon, sugar as the backbone. Here we're looking at a DNA nucleotide. The sugar that we use is the deoxyribose sugar. So DNA and RNA nucleotides are going to use a different sugar. That's the big difference. Deoxyribonucleic acid uses a deoxyribose sugar. In a minute, I'll show you an RNA nucleotide which uses a ribose sugar. Right? So the sugar is going to be different between DNA and RNA nucleotides. So here's the sugar with the carbon backbone. Attached to it is going to have a phosphate group. Now, phosphate group is nice and polar and charged. And then, so each of your nucleotides are going to have a sugar and a phosphate group. Every single one has sugar, phosphate, sugar, phosphate, sugar, phosphate. The part that's different is their functional group, the reactive part of the molecule. The functional group in a nucleotide is called the nitrogenous base. There's 4 types of nitrogenous bases. We'll talk about those in a second. Right. Good? So far? Nucleotide structure. All right. So when we take a look at a nucleotide, again, here we're looking at a DNA nucleotide. It's got a deoxyribose sugar. Here's its phosphate group. What's going to be different is the functional group. Right? We have 4 options. Right? Notice how we have purines and pyrimidines. Right? Their size is different. Notice how purine has 2 rings, pyrimidines have 1 ring. Right? So 4 categories of nitrogenous bases. We oftentimes call the nucleotide or name the nucleotide based on which nitrogenous base it has. So the nucleotide that contains an adenine nitrogenous base, we just call it adenine. A nucleotide that contains a thymine, we just call it thymine. Right? A nucleotide that contains cytosine, we call it cytosine. Right? And guanine, of course, guanine. Right? And sometimes you'll see them abbreviated with letters, right? Just the first letter of the nitrogenous base, right? But each nucleotide is going to have 3 components, all 3 components. Now, if we take a look at DNA, DNA is actually going to be 2 strands of nucleotides that are interacting with each other, binding with each other. Now, we see DNA illustrated like this, like a ladder that's then been twisted, right? The backbone, what we call the backbone of DNA right in here, is a sugar phosphate, sugar phosphate, sugar phosphate. The sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphates are the part of the nucleotides that bind to each other. And then they're going to project their nitrogenous base towards the center of the molecule. And then notice how 2 nitrogenous bases will form little hydrogen bonds between them. So if we take a look at this diagram, you can see AT, 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 AT. Adenine and thymine are always going to bind to each other and form a little bond. Right? Guanine and cytosine, if you take a look, are always going to bind to each other. We call this the complementary base pair rule. The way I remembered it was cytosine, the curvy letter, always binds with guanine, the curvy letter. Adenine, the 2 stickies, right, always binds with thiamine, also straight letters. Yeah, I don't know. Maybe that'll work, maybe not. Does this make sense? So DNA is a double strand of nucleotides held together by interactions between the nitrogenous bases. The sequence of nucleotides is what's going to store information. But I'll come back and talk about that later.
I have a question.
Yeah.
On the last nucleotide pair, or I guess nitrogenous base pair diagram, there weren't any uracils. Is there a reason for that?
Yes, because uracil is going to be something we only see in RNA. So right now we're looking at DNA. I'll get to uracil in a second.
Okay, I remember that now. Thank you.
Great question. All right, so here you can see the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar backbones, right? And the nitrogenous bases are in the center. Again, the nitrogenous bases are going to form little hydrogen bonds between them, right? Here you can see guanine and cytosine, thymine and adenine, yeah, holding the 2 strands together. So DNA is double-stranded. It's longer, much longer than RNA. And it uses that deoxyribose sugar. If we took a look at RNA nucleotide, we would see that it's got the phosphate group, it's got a sugar, but it's a different sugar. It's a ribose sugar. So the nucleotide is slightly different. And then it's going to have a nitrogenous base as well. 3 of the nitrogenous bases are the same as what you see in DNA, right? You've got adenine, you've got guanine, you've got cytosine. But thymine, you substitute for uracil. Right? So if you're looking at 2 nucleotides, if you can assess the sugar, you can tell whether it's a DNA nucleotide or an RNA nucleotide. If you're looking at a strand of nucleotides, right? If it's a short strand and it's a single strand, RNA just forms a single strand, you know you're looking at RNA. The other big ding, ding, ding is if you see uracil. Uracil is only found in RNA. And it's going to bind, complementary bind to adenine, but I'll get to that later. It's a different lecture. Cool? I think that's my last slide for this PowerPoint. We'll have 15 minutes. All right, let's see. Oh, have I been shut? I could have sworn I shut that down. Any questions on organic molecules? Okay, so for your exam on Tuesday, you have the introduction lecture, scientific inquiry concepts. We covered chemistry. We covered molecules. organic molecules and inorganic molecules. So we talked about water, and then we just finished up talking about the 5 organic molecules. You've got the patterns in science little part of lecture that's on Canvas as a homework assignment. Yeah, so everything through right now will be on your exam. I'm gonna start reviewing, introducing cells. The cell content won't be on this exam. It'll be unit 2, right? But I'm gonna go ahead and start introducing it. We've got a little quick recap poll, checking understanding. Ten more, go, go, go. Last call. Okay. Going once, going twice. All right. Here we go. All right. So the bond that forms between, oh, I didn't typo, forms between amino acid side groups, the functional group. What do we call the bonds, the level of protein structure that refers to the bonds that form between the side groups, holding the protein structure together. I shouldn't have closed out that other PowerPoint. That's tertiary structure. Remember that primary structure is the sequence of amino acids, which amino acid you put next in that growing chain. Secondary structure is the natural pleated sheeting that happens as you form bonds between carbons, and the spiraling that's caused by hydrogen bonds. Tertiary structure is the interaction between the functional groups, the side groups. Holds the 3-dimensional shape together. Better? Quaternary structure is taking a look at subunits of proteins that then come together to work as one functional group. Make sure you go back and review protein structure because it definitely didn't work. If I can find a video, I might send you guys a little video with an animation or something. Cool? Any questions on that? Okie doke. Are you ready to start learning about cells? So I'm going to hopefully just get through cell theory today. I'm going to introduce cells and talk a little bit about what we know about cells. And then we'll come back and take a look at the different categories of cells that we see on Earth. We're going to spend a lot of time talking about eukaryotic cells because that's what we're made out of, right? I'll do a little comparison between plant and animal cells, and we'll talk about cellular structure, the major organelles or operational units inside of a cell. So this we'll do next week. I just want to see how much we can get through today in our last 10 minutes. All right. So one of the things you want to be familiar with is that cells are really little tiny bitty things, right? You can't see them with your naked eye. If you looked at the head of a pin, right, there would be thousands of cells on there. Cells are really small. The largest human cell is a human egg, and you still would struggle to see that with your naked eye. if we took a look at Shamu here, a big orca, right? Even though it's a big organism, it is still made out of itty bitty tiny cells. All cells are small. Right? They're all about the same size-ish. Orca just happens to have more cells than you do. You have about approximately a couple trillion cells in your body, human cells. That's outweighed by the number of bacteria cells that you have in your body. So if you've listened to the Patterns in Science lecture already, right? We talked about scale, right, or perspective. Right? We started off the semester at the small scale, looking at atoms and atomic structure. We put atoms together and learned how they form bonds to build small molecules, right, like an amino acid. And then we zoomed out and talked about how we can use dehydration synthesis to put small molecules together to build macromolecules like a protein. What we're gonna be learning next is how we can put together a bunch of those macromolecules to make an even larger structure like an organelle, a structure that's inside of a cell. And then we're going to put a bunch of organelles together to understand how cells operate as a whole. Right? We're going to be focusing on eukaryotic cells, which notice are significantly larger than bacterial cells. Yeah? Once we learn about cells, then we're going to come back and start talking about organ systems and whole bodies and how they interact with the environment. Right? But we're still down at the pretty small scale of cells. So why is it that cells are small? It has to do with exchange rates, right? It turns out that the smaller you are as a cell or the smaller you are as an object, the more surface area, outside area that you have relative to the volume inside. We refer to this as surface area to volume ratio, right? The smaller you are, the more surface area you have per unit of volume. Right? And it turns out that that's really important. As a cell, you have to be really, really, really small so that your surface area is sufficient to exchange gases and nutrients with the environment enough to support the contents, the volume of your cell. Surface area to volume ratio is really important, right? Really, really important. Cells are going to be moving a lot of their material by a process known as diffusion, and it's only efficient at really, really, really small distances. Right? So there's a few reasons why cells are small, and cells are universally small. Right? There is no organism that has big megacell. Well, that's not true. There is one weird organism, but it's got some crazy adaptations that I'm not going to talk about. Good. Surface area to volume ratio. All right. The other thing before we walk into cells, you got to see the little GIF at the beginning. You know, we walk away from intro bio class with this idea of what a cell looks like, and we think that all of our cells in our body are the same. They are not. Your cells are going to exhibit patterns of form and function. Right? They're going to be designed. Both their structure and their contents are going to revolve around the function that they're performing. Right? So while you have trillions of cells in your body, they're not all the same. Right? We've got about 200 categories of cells, but even within each category, there's diversity. You can be taking a look at a columnar cell in the digestive system and a columnar cell in the gallbladder, and they look different from each other because they're performing slightly different functions. So if we take a look at this illustration, right, very quickly kind of gives you an idea of some of the different shapes that we see. So here we're looking at a neuron in a brain. A neuron is a cell. And a neuron, while it's a eukaryotic cell and it has a nucleus, notice how it also has all these branching cellular projections. That's because it has to connect and communicate with other neurons. Right? And the more branching it has, the better it can process information. Versus if we look at a muscle cell, so here we're looking at a smooth muscle cell. This is a type of unconscious muscle you have building your, lining all your organs. It's long and narrow so that it can maximize contracting and producing motion. Sperm cells, streamlined shape, long flagella for being able to maximize swimming and moving through the female reproductive tract. All cells exhibit patterns of form and function depending on what they're doing and where they are. They're not all the same as the blob that you're going to see in the textbook. All right. So cells, what do we know, right? Cells kind of interesting. We're still learning a lot about cells as we get better and better technology. It turns out that our understanding of cells has always been limited by technology. And it's not until the first magnifying object, the first microscope was created, that we were able to identify the cell. And that was way back in 1665. Good old Robert Hooke got this. The first microscopes were just these magnifying spheres, right? And of course, as a good scientist, he started to look at all kinds of different things under his scope, bodily fluids, objects that he was collecting from nature. One of the things that he put under the magnifier was a little sliver of cork, right? And cork like you would have in a wine bottle. Cork, remember, comes from a tree. It's a bark of the cork oak, right? So it's a little core of that bark. And so it's made up of cells. And as he was looking at that cork underneath the magnifying glass, he saw all these little chambers. They reminded him of jail cells. So he called them cells. Right? So cells are named after jail cells. I'm interested to know the story of how Robert Hooke was so familiar with jail cells, but that's a story I haven't been able to find yet, right? But cells. And ever since then, we have been developing better and better magnifying tools, right? And since then, we have added more and more information of understanding cells. And again, even now, as we get better and better technologies that allow us to see really, really, really small things, we're learning all kinds of new subcellular structure all the time. It's kind of fascinating, right? There's still a lot of undiscovered knowledge in pertaining to cells, right? But here's what we do know, right? What we do know has been combined into cell theory, right? So again, theory, big, big idea, a collaboration of a lot of knowledge that has been tested and vetted extensively. So we feel that we, kind of have a good grasp on it. Here's what we know about cells. And are you going to want to know the summary of cell theory? Yes, not for Tuesday, but for the following exam. All right, so all living things are made up of cells. Every living organism has at least one cell. Right? Unicellular organisms dominate life on Earth. They're the most abundant, right? But they are made up of cells, a single cell. Every living organism is made up of cells. Right? They are the structural and functional unit of living organisms. That means that they're the building block for how to build the structure or the body of an organism. They're also the functional unit where everything that keeps a living organism alive happens is inside the cell, right? So here's some crazy things to think about. All cells have to come from pre-existing cells. All cells have to come from pre-existing cells. You can't make a cell out of nothing. You can't build it from scratch. All cells have to come from pre-existing cells. But my mom made me. She didn't make it from nothing. It wasn't magic, right? There was a pre-existing cell, an egg, that was fertilized and then divided. All cells come from pre-existing cells. You have to copy and divide a pre-existing cell to make a new one. And one of the reasons why all cells have to come from a pre-existing cell is because they contain your heredity information. In other words, it contains your DNA. It contains the code for how to build proteins, how to cycle ATP, how to build plasma membrane. Right? And you can't reassemble that code from nothing. You have to copy a pre-existing strand of DNA to put in every single cell. And this is an important driving force for why every cell has to come from a pre-existing cell. You need a strand of DNA to copy in order to put into the new cell. Virtually every cell in your body has your full set of DNA. All cells are going to have nucleic acids. There might be a couple exceptions, but I'll get to that later. Good. All cells come from pre-existing cells. You copy the existing DNA and organelles, and then you divide those cells to make 2 cells where there was once 1. We'll talk about cell division in a couple weeks and learn about the 2 types of cell division, mitosis, meiosis. We're going to talk about how to copy DNA, right, when we get to DNA lecture. So we're going to spend a whole unit 2 inside of the cell and learning how cells operate. All cells are pretty much the same, whether you look at a sea star or a human or a plant cell. We are all basically the same. The phospholipid membrane and the types of proteins, right? The organelles that we have inside that are keeping our cells alive, right? All things are pretty much the same. Remember, we share all that fundamental DNA for how life functions with organisms, all organisms on Earth, right? You learn that we share 70% of our genes with a sea star, share 40% of our genes with a wine grape. All right, and then all the function that keeps you alive happens inside of the cell, right? So all of the energy flow, all the energy conversions, your metabolism, right? Your cycling of ATP, all that happens inside of the cell. If you're a plant, you also have photosynthesis happening inside of your cell. Ooh, I've got to stop there.